And I have a question for Dr. Amy Ramsey. Thank you for the nice talk. And you mentioned before that one of the two mice model, the mutation was both loss of function and gain of function.
Yes.
Can you explain more about this? What does it mean?
Well,
thank you.
It started with the characterization in HEK cells that was done by CFERB and at Steve Trinellis's group at Emory, where they were looking at agonist potency, all of the different domains that they look at, and it looked like it had increased agonist potency and no change in magnesium inhibition. It was increased potency for both glutamate and glycine. So that would have been a predicted gain of function, but they also saw some reduction in the trafficking to the surface. And I think there was another group that recently published a similar study and reported the same thing with a big deficit in trafficking to the surface. So it does seem that there's a problem with trafficking. Then my collaborator, Evelyn Lamb, was studying by doing whole cell recordings, was studying these brain slices from these mice. And when she bath applied NMDA, there was a massive current. So again, it was consistent with the gain of function. But then when we collaborated with Graham Collingridge's lab, they were looking at kind of synaptic NMDA receptors and AMPA receptors in the hippocampus, doing field recordings. And there was a reduction in synaptic NMDA current and in LTP, like really dramatic reduction in LTP. So now, it's complicated, and it's probably that trafficking piece that is explaining it. And I think it has, there are other receptors at work, or other channels, ion channels at work, besides just the NMDA receptor in the whole animal. So I think that it's really shown me the importance of making the animal with these complex ones. I think that there's somewhere it's really straightforward. It's a clear loss of function, but then there's others where I think it's complex. Yeah.
I'd like to thank all three speakers for nice talks. I have a question to Amy, where I was very convinced that the NMDA receptors are expressed in higher levels outside the brain is pancreas, beta cells, and I would expect, or I was thinking using some form of tests concerning glucose level and so on to be used as to look how well you can treat or help in
those
first.
biomarkers.
So what are your results concerning glucose?
Yep. We have a big program on that. So I'm actually really excited about your interest in the GRINs as metabolic disorders, because we definitely see reductions in the amount of I'm trying to remember what it was, the alpha cells or the beta cells. I think it was the alpha cells actually. But definitely the size of the islets is much smaller. Actually, I don't remember if she did this staining for alpha versus beta. So all I know is the islets are much smaller in the Q536R and the knockdown mice. So the loss of function. And yes, they have kind of higher levels of glucose. So it's kind of looks like a bit like diabetes, actually. Yeah, so there's definitely a phenotype there. And yes, blood glucose could be actually an even easier biomarker than the CBCs. Mm-hmm. Yeah.
Okay. I have a short question for all three of you. Dr. Tardio, I think the answer to this is going to be no, but are you able to share which type of modulator you were using?
Sorry?
The modulator you used, the PAM.
Yeah.
Are you able to share what it was?
Not right now. I'm sorry.
I thought I'd try.
No, but soon. So we have mostly completed the preclinical and these proof of concept clinical studies. So we will submit the paper very soon. And since there are also some pharma partners involved, we cannot.
Understandable.
Sorry.
That's okay. Thank you. Dr. Ramsey, do you think the iron deficiency could be tied to sleep disorder? Sorry about that.
To sleep.
Yeah.
I am not familiar with the literature, but I know that the mice do have a disrupted sleep. So in their normal sleep cycles, they are more active in their home cages. But I think what would be interesting is to kind of push the mice either by giving them iron supplements or by giving them something that would chelate some of the iron. It's not clear whether the problem is too much iron, believe it or not, because too much iron can then lead to iron deficiency. So if too much free iron, because the free iron can destroy the cells that store the iron. And so we're not sure yet what's going on. But I had not thought about this connection between iron and sleep.
And then finally, Dr. Hudac, thank you, Dr. Ramsey.
Mm-hmm.
You have the slide up here. I'm just wondering, is this study going to be open internationally?
Yes. So the reason why we put it together yesterday, realizing it might be helpful to just kind of have it be available. So it's in English, first of all. So that's our limiting factor at this moment. We loosely translated some of the measures into Spanish, expecting we would have potentially more Spanish families here. My RA who speaks Spanish did this translation, so it's not validated, but we thought at least this is just pilot data for us. So anyone's welcome to do it. We might have some kinks because, as I said, we opened it yesterday. So if you have problems, just come find me and we'll work through it because there might be some bugs I have to fix. But we're excited. Any age as well. Depending on how old your kiddo is, then you might have one less measure to do.
Okay, thank you.
Yes. Okay. I also have a couple of questions. For the first speaker, if you think about using the PAM, can you comment something on how you perceive whether the deficit is more present in excitatory or inhibitory cell lineages, and whether you're actually curing a feed-forward inhibition problem or a prominent deficit in main neurons?
Yeah, this is a good point. Very complicated. So nowadays, I think that there is no pharmacological way to target a specific NMDA receptors on a cell type manner. And this is something that we're facing all the community. So we only see the global effect on the combination of this effect on excitatory and inhibitory. So unfortunately, we are not in the position to really try with this PAM and then to evaluate. Well, we could evaluate doing single cell patch clamp, whether this treatment in mice is increasing or modulating inhibitory neurons, but we only have the global effect.
But could you, because in the first slide you showed this computational model with all these assumptions about the effects of gain of loss of function through different genotypes, but would you think there would be a way to combine the computation modeling to infer whether there's more prominent effect on inhibitory or excitatory lineages? And if there were, what kind of inhibitory cells that would be?
Theoretically, yes. If we use computational tools and artificial intelligence, we can integrate and we have a precise model of what is the transcriptomics of the different cell types and the expression of the different subtypes of NMDA receptors in all the neurons. Potentially we could, yes, but we are not in that position yet.
Yeah, interesting. And about the iron, I was just curious whether you were actually hinting that you think that at the end of the day, that apart from all the neurological phenomena that they were studying, that you may think that either in development or later on, this blood cell problem, this energy homeostasis or iron homeostasis problem, is actually also contributing to energy expenditure in the brain and might be actually a proximal cause of the neurodevelopmental phenotypes? Is that what you're getting at?
I would like to answer that. This is a question that I have. So I mean, I think we've done a lot of things where we've manipulated the levels of NMDA receptors in different cell types. And I think at the end of the day, the neuron is the most important cell. But also there's a lot of adaptations that the brain can do to, to compensate. So I'm wondering if we can help the brain do what it does by, but the brain needs a lot of energy. So I think if there's an energy problem, the brain is always the one that's going to suffer from it.
Exactly.
So delivering energy, I think is essential and could be really beneficial. And so I think that an energy problem could be secondary to the fact that the neurons are struggling, that they're working harder, that they're firing more. But I think it's it could be certainly not helping if there's a problem delivering energy.
Potentially, you could study another very unrelated genetic disorder to see whether the same homeostatic problem occurs irrespective of of NMDA, for instance. That could be a thought. Yeah.
That's an interesting idea. Yeah, that's a good point.
Okay. And the last small question to Caitlin. You have done a lot of work on EI using FOOF. Are you planning to study the interaction between EI and oddball deviations as well, or have you done that already?
Yeah, so we have resting state as well on these kiddos. So we are planning to look at, so you can use some software to look at excitatory versus inhibitory contributions that at each child, so they might have their own kind of EI balance traits. So we plan to do that. We also, as you probably know, Aneta and Laura are working. So I presented on the pan-neuronal knockdown, but we also have the excitatory and inhibitory subtypes, neuronal subtypes of the fly. So we can use them to look more specifically at behaviors, sleep, and habituation. So kind of pairing the two, both with the FOOF approach, is our goal for sure.
Nice. Thank you.
Yes. So thank you so much for the presentations. They were very good. So it's a question for Amy about iron well, I think that from a practical point of view in children, it will be very challenging to test iron as a biomarker because iron tend to be a little bit vulnerable during growing and development. So it is very common to find ferropenic anemia in normal kids.
Okay.
And we find it quite frequently in many patients with neurodevelopmental diseases, neurological diseases, because they don't eat enough, they are growing a lot, and sometimes it is just due to that. So unless you have like very low levels or very high levels, something that is really very different to what we found in normal situations, it will be really challenging. Or maybe you find other biomarker that is not exactly iron or ferritin, but it's linked to iron metabolism. So it is something to look at, and we can do that like very quickly in our patients because we have all these nutritional patterns and nutritional, values in our children, and see if this is very low. We found very low levels of free iron in a particular energy deficiency disorder and an inhibitor of metabolism. We are checking that. So that was the first thing. The second thing is that energy metabolism is impaired, of course, in NMDA disorders, and it has been demonstrated that there is a downregulation of glucose transporter in the brain.
Yes.
Yeah, and we found that in other channel disorders, and they may also mimic GLUT1 deficiency. Sometimes glucose levels are a little bit lower. And so it has been also recently described a relationship between some parameters of energy metabolism and iron metabolism because of a signaling pathway that finally will download the levels of iron. So maybe if you go to this link between energy metabolism and some monocarboxylate transporters that finally lead to this depletion of iron, it's something that you can explore maybe in the mice. But I think that we could just start by looking at iron metabolism in our patients and see if these values are really different to what we see in other neurodevelopmental patients. That
that of course would be very exciting. Thank you for that. Yeah, actually, it was we were the ones who found the GLUT1 reduction in the knockdown mice. So yes, there is also 2-deoxyglucose uptake is impaired in the mice. So for sure, there's a problem with glucose homeostasis as well, that there's too much glucose in the blood, not enough in the brain, because it's not being imported. And there could also be some problems with insulin and glucagon release. So yeah, but I really appreciate your perspective about the iron metabolism and homeostasis, and especially in pediatric. Yeah, thank you. So exciting.
Quick question for Xavi.
Yeah.
Very interesting data that you show on the PAM, the new PAMs in the model. Did you test L-serine or D-serine in the model? And if you did, what did you see?
In this mouse model?
Right.
No. This is something that chronologically, so we jumped directly from in the L-serine studies from the cellular models directly to this an initial case that was published in 2019, and we didn't need to do this L-serine on GRIN2B mouse model. This is something that is pending. So somehow we would like to compare what is the effect that we observe on L-serine in this model and to compare with the PAM effect. But for a matter of time and resources, we didn't.
Yeah, thanks.
Just a question about the EEG net. Is the data all kind of relative to that patient? You have a baseline and then you're looking at changes, or is there any inherent differences, maybe in individual electrodes or individual zones you find different with the autistic or the GRIN2B patients than you find, say, with a healthy control?
So in a traditional study, everyone would be in the lab and we'd have controlled noise. You saw I had these kids in every nook and cranny of houses, right? So we've adapted analytic strategies to basically look at within person change. So there are some, and that's I think one of the biggest limitations is that like the baseline processes are going to have to be kind of normalized for each kiddo. So we plan to kind of work on that post hoc on our kind of back end. So it's hard. Yeah. So it's harder to compare one child to one child. We're really talking about the attention effect for kid A versus kid B. But that seems to be, it's a good start, right? So if we were to go towards clinical trials, I do think that we would have to kind of think through, maybe normalize things even a little tighter. The home was just such a great environment. Kids are happier. I was telling like their dogs are around, their favorite toys and snacks. I've never had so much behavioral compliance, particularly from this population before. So getting them out of the hospital and into the house was a huge, huge just improvement in terms of the amount of data we could get. And most kids got through all 20 minutes, 25 minutes. So.
Okay, I have one last quick question for Dr. Hudac about the fly model and the pros and cons, because glutamatergic synapses are at the neuromuscular junction. I mean, they're very different. So why the fly?
So in part, I think because we really are kind of starting to look at this inhibitory excitatory piece, and being able to control those neuronal subtypes is a true advantage. This is a group, they've been doing this for a long time across a variety of different disorders. So like GRIN wasn't their primary focus. So if we were we're looking for an animal model for GRIN, I don't think we would have started at the fly, but we can still use that information as a mechanistic understanding. So I think the idea would be to kind of start to expand. I think habituation, attention, learning are going to be particularly important to look in our other species. And there's probably other behavioral assays that I just am not less familiar with as a human person. So I think it's just one piece of a big puzzle.
Yeah, we got some questions online as well. I'll ask you, Dr. Xavi, later for one because it's a longer one and maybe you need to read up on it because it's regarding a different study that you could collaborate, but that will come as a direct question to you. You can come over here. But the quick one for you is from Alan. Will the study with PAM evolve to a larger clinical study? If yes, what will be the schedule that you currently look for?
Yes, well, this is a question for me and also for Angels for the clinical studies. So basically, with this proof of concept study, we will try to publish in the next month. So hopefully at the end of the year or in the next 6 months. And once we get this publication, we are also working on a clinical study with clinicians in San Juan de Deo, mostly, and that are leading this part. And they are working already on the ethical committees, local ethical committees, and all the regulatory aspects also to get some funding that is required for all the neuropsychological assessment that requires a specific team of neuropsychologists that can lead and can dedicate time for the follow-up of the patients. But the idea is to do the same as for Elserin trial.
Thank you for that. And the second one was also for you, and it's I'll read it out, and you can answer later if you need. It's from online as well. It's I'm curious to know if you are familiar with the work conducted at Marinka Žitnik lab, particularly the Shepherd and Therapeutics Data Commons TDC model. Given the open source nature of these models, do you envision the potential for integrating such tools in your current research endeavors to further the understanding and treatment of GRIN-related disorders?
I think that I'm not familiar with these works, but I will read the question perhaps later on.
Yeah, we'll look at it directly. Yeah,
thank you, Alan.
And the last question I'll keep for the next session because it's an open question for everyone, and it's the answer is I think the discussion it's on gene therapies, but that's a longer discussion. I think when we need to stop for the lunch.
So before we go, I really would like at one point to maybe take it as a topic for the roundtable discussion, this topic about the hypoglycemia, because this is an issue that I think it needs a little bit of a, not urgent, but maybe a little bit urgent topic. Sorry.
It's not exactly well, it's not exactly hypoglycemia. Glucose levels are normal. They are not high or low. Well, I mean, if it is something, it's in the brain, at least what has been demonstrated by now, and it's a tendency towards neuroglycopenia. But I mean, and it is also, it has been demonstrated mostly in the animal models. Also, we have found in some patients in the CSF, but it is not something that is constant. I mean, what it is clear is that energy metabolism is involved, and we don't know exactly all the pathways and all the mechanisms of energy metabolism. But well, this could be also something to approach from a therapeutic level. Yeah.
Comment. I think a lot of our kids do have issues with glucose regulation. And I think the question was more related to the reactive hypoglycemia, because it seems that a lot of our kids can have like these drops in energy, like all of a sudden without no further explanation. So, but let's keep that for the roundtable discussion, because this is really like interesting topic. So,
and talking about hypoglycemia, let's go eat.
Let's go eat.